let's continue with a review of lipids. So lipids here, I have a picture here that hopefully will sort of call to mind some of the things we've talked about already with lipids. But one of the things that's sort of the bottom line with all lipids is that lipids are hydrophobic. So most lipids that we're looking at, we will consider things which do not like water. And therefore they are non-polar. They're made up of a lot of hydrocarbons. So remember, it'd be like chains of carbon with a lot of hydrogens around them. So let's take a look. So the examples of macromolecules when we're looking at lipids are the list we see below here. So as categories, we can see fats and oils, which we sometimes put under the umbrella of the triglycerides, waxes, phospholipids, which we spend quite a bit of time talking about and will again, and then steroids. But why we need lipids, other than the more obvious thing about our cells and cell membranes, is that lipids provide us with a lot of energy. They give us long-term energy stores, and they also help insulate us, as we all know right now. If you're feeling a little chilly, we need the lipids. We need fats that are a part of our body tissue. They also serve as a building block for hormones. So if we don't have enough lipids, then we're going to have some problems creating certain hormones. And we mentioned the cell membranes already. We started with fats and oils. We would see the structure of fats and oils. We can see the glycerol as the backbone with these fatty acid tails. Remember that a fatty acid is this long chain of hydrocarbons that has at the top of it, right here, has a carboxylic acid. So this COOH right in here, if you look at the functional group list, you'll see that's a carboxylic acid, and that's a part of this fatty acid structure. And when this fatty acid then joins with glycerol, as a matter of fact, if three of them do, because they join at these particular, at these hydroxyl groups, these OHs on the glycerol, what you get, and they join right here, where you can see the carbon, oxygen, carbon, this thing right here is called an ester linkage. So again, dehydration synthesis, we kick out water, and we end up with these bonds that form, these covalent bonds that form, and this particular type of covalent bond is called an ester. So we have ester bonds. We have three of them here. And that is either called a triacyl glycerol, or we can just call it a triglyceride. We've seen both of those terms. Now, they've saturated here. So here's the fatty acid chain. Here's just one of them. Here's on this. Instead, we flip this. So here's the carboxyl group on this side. You can see that this one is a straight chain, and that there are all single bonds. So when we look at the single bonds, and we're just talking about the carbon-to-carbon bonds here, then we're talking about a fatty acid chain which we call saturated. It means that it has the maximum number of hydrogens that it can have because there are no double bonds. And the reason saturated fats are an issue with us in our diet is because their structure, this linear structure with no double bonds, nice and flat, will allow them to pack, and they pack tightly, and they can create things in our bodies which kind of clog our blood vessels. So that's why we try to avoid saturated fats. This fatty acid itself is called stearic acid, so it's just one form of this kind of saturated fat. But here's oleic acid, and this one does have a double bond, so you can see it right here. And therefore, you can notice that it looks like there's something missing here. We have fewer hydrogens. We have two fewer hydrogens because of the double bond. So this one would be called a monounsaturated fat because it has one double bond. And polyunsaturated fats are ones that have lots of them. But either way, unsaturated fats are considered oils. So they aren't solid at room temperature. They have this ability to have more flexibility when they have the double bond. Now, what you see on each side of the carbon to carbon double bond tells us a little bit more. So let's go into that. So if we see in this example above, here's a saturated fatty acid, and here is a type that we're calling the cis form, meaning that the hydrogens that are attached to those carbons that are double bonded are on the same side, then that is the cis form, and cis forms have this kind of bent structure, so a little bit of a kink in the molecule, which means that these can't pack tightly with other fatty acids. When hydrogen instead, though, is across the double bond, we call that trans. And it's the trans fats that are not particularly good. We want to avoid. Let me move me so you can see. They will increase what are called the LDL cholesterols, which are the ones that are bad for our heart. So we don't want the low density lipoprotein cholesterols. That's what we don't want. Okay, so just think LDLs. But anyway, we don't want trans. We like the cis. We like the cis, because they have this bend. There are other, though, types of fatty acids, and this is kind of a different model that you can see here. So remember that you assume that at every point of each one of these lines here are carbons. So this would be, again, a nice long carbon chain. Some double bonds in here. You can see that's a double bond. This is a double bond. This is a double bond. So there's 3 double bonds in this structure. And we have, as a consequence, you can see it's kind of very much, it's very kinked or very curved. This is an omega-3 fatty acid, and it's found in fish primarily, and we can find others that are called omega-6s, and these are the ones we like. So they're essential fatty acids. We want to get them in our diet because they're healthy for us and they do good things. So there you go. Waxes, we didn't spend much time at all looking at waxes other than to say, and so I pulled a picture up of something that, I'll move this over here. You can see that waxes are, they have, again, carbons, a lot of hydrogens, some oxygens. They also have a little bit of an alcohol component to them. But the purpose of them really for waxes, the only thing we wanted to know is that because they're hydrophobic, they're going to prevent water loss from the surface of plants and also from birds' feathers and things like that. Steroids have their own structure. They have these sort of interesting ring-like structures. And these are important for a lot of things. They can serve in producing hormones, or they can act as hormones, and they also are involved with keeping sort of the fluid nature of the membrane. Think cholesterol. Here's the classic picture of a phospholipid where you see here's the phosphate group. Remember, functional group right here. There's a lot of negative charges on it. Here's glycerol. But glycerol in this case only has 2 fatty acid chains that are attached, one of them being a saturated one. You can tell because it's straight, one being unsaturated. So this helps the phospholipid when it's with a whole group of them to also be, to not pack together too tightly, so that it allows a little bit more fluidity to that membrane. And you can see this is just some different types of models that we can look at with the structural formula. And then this one that looks like a toy of some kind. And then this was the typical one that we draw. So it's usually at least one of these tails is bent and one is straight, but this can vary a little bit depending on what climate things live in. The term amphipathic is important just in that it helps us understand that the amphipathic means that this molecule, this phospholipid structure, has a part of it that likes water and a part of it that doesn't. So that dual nature is shown here, where we know that it's these tails, which are those hydrocarbons, are the part in here that don't like water. They're hydrophobic, they're nonpolar. But out here, the phosphate heads are hydrophilic, and therefore they're polar. And so that helps these arrange in a particular way. Also note though, that these are, while they're right next to each other, they aren't bonded together. So each one of these phospholipids, they arrange themselves this way spontaneously. If I took a whole bunch of them, shook them up and threw them in water, what they do is exactly what you see here. So you would see them form these usually kind of circular, looking like a cell membrane, with the tails on the inside facing in toward the away from water, and then the heads outside. Here's a picture of steroids again. Here's cholesterol. Here's another steroid that some of you may be familiar with, cortisol. This is a steroid that is released in response to stress. So cortisol is released and then activates different parts of our body that are dealing with stress. Again, hydrophobic, insoluble in water, and kind of important. So that's it on the lipids. We'll go back to proteins in just a minute.